I'm Professor Rausch, and I'm an assistant professor in biomedical engineering, aerospace engineering, engineering mechanics, as well as the Odin Institute of Computational Engineering. So what does the Rausch lab do, or the soft tissue biomechanics lab? Our main focus and interest is on studying and understanding soft tissue disease, of course, ultimately with the vision that we could change the lives of many patients that suffer from these diseases. We do that by using multiphysics experiments, numerical modeling, microscopy, tissue level mechanics, in situ imaging, and you can see it here just above the mouse, but we also do mouse modeling in large and small animals. Specifically, we're really interested in right cardiovascular disease. As I said, we use a range of different techniques and tools to do that, but we ultimately do basic science toward predictive modeling. And I'm going to use my window and move it a little bit because you can't see that right now. And the vision here is to improve diagnosis, improve therapy. Important though, we're not a modeling lab. So about 50% of our effort goes into the basic science to inform modeling, and about 50% of our effort is on the actual modeling side. My current lab consists of 4 graduate students, 2 BME students, 1 aerospace student, and 1 mechanical engineering student. And my student cohort essentially reflects the interdisciplinarity of my own training, of my own interests, as well as the lab. So what is the forgotten side? Well, the forgotten side is what we often call the right side of our cardiovascular system, because it really hasn't been studied for decades. And much of it was forgotten, and the side itself has been forgotten until recently. So what is the right side or the forgotten side comprised of? The right ventricle, the tricuspid valve, and the venous vasculature. And we specifically focus on those aspects of our cardiovascular system. So I'm going to move my window again and maybe reduce it a little bit in size. Here we go. We have 3 main research thrusts, one on right heart mechanics, one on the tricuspid valve, and one on thrombus mechanics. We also have other projects related to medical device design, as well as other basic science questions, such as thermomechanical coupling in skin. But I'm going to talk about these main thrusts today. So first of all, right heart mechanics. First of all, what is the right ventricle? You know, the heart has 4 chambers. It has the left ventricle, the right ventricle, and the 2 atria. The left ventricle is being studied by a ton of different groups, and there has been a lot of work, but not on the right ventricle. And so the right ventricle is really comprised of the right ventricular free wall, as well as the septum that is shared between the 2 sides, and the papillae muscle. And we talk about the tricuspid valve later on a little bit. So why study the right ventricle? Why are we interested in this? Well, first of all, little is known about the right ventricle's function, structure, or disease. And that's an issue because there's a ton of different diseases and disorders that affect patients related to the right ventricle, and we just don't understand it. One of the ones that I'm really interested in is a phenomenon in which we implant left ventricular assist devices, LVADs, to save patients that have left heart failure. But in 20% of those patients, once you save their left ventricle, actually their right ventricle fails, and we don't understand why that is. It is very likely that wall stress could be a predictor. And if we were able to predict in whom that happens, then we could, of course, take special care to prevent, in those 20% of patients, right heart failure. Very importantly, we need more basic science, and we need high fidelity computer models to understand right heart failure and to understand this phenomenon. So in my lab, what do we do to fill this gap in knowledge about basic science and about computer models of the right heart? We use a large animal model. So these are surgeries that we don't do in my lab. We have a collaborator who does this, and we do surgeries in sheep to induce right heart failure. And then we do custom mechanical testing. So we developed both protocols as well as actual equipment to test the right ventricle in mechanical modes that are relevant for the in vivo function of this tissue. We use MRI, both sort of standard anatomic MRI as well as diffusion tensor MRI. You see here a video of the right ventricular free wall under diffusion tensor imaging, where you can see the macrostructure as well as the fibrous microstructure. We also use histology, and we do use finite element modeling. So here you see a pipeline where we go from animal data to sort of 3-dimensional finite element models of the right ventricle. Our second thrust is on tricuspid valve mechanics. You already saw this. The tricuspid valve, of course, is a fundamental element of the right ventricle. It's the check valve that prevents leakage from the right ventricle into the right atrium. So we want forward flow, not backward flow. And that backward flow would be called leakage regurgitation. The tricuspid valve, as the name says, has 3 leaflets. It has an annulus that basically describes the interface between the valve and the surrounding myocardium, and has chordae tendineae that attaches the valve to the right ventricular myocardium. And so you see these are intimately interconnected. So studying one without the other is problematic, and that's why we do both. But why study, again, the tricuspid valve? Well, first of all, again, this may look familiar, little is known about its function, structure, or disease. Which is shocking because about 1.6 million Americans have severe leakage. And that means these patients are exercise intolerant, they're short in breath, and the quality of life is relatively low. In many, it's not repaired because we don't really understand what's going on, so it's hard to repair. But when we do repair it, the repair fails in 10 to 30% of patients within a few years, and we don't understand why. And so again, we need more basic science and high fidelity computer models to optimize repair, understand what's going on, and to improve our current standard. What tools do we use to do that? We use large animals, again, sheep. We're about to start actually in small animals, rats as well, and we use human data. On the right-hand side here, you actually see a human heart. So this was a heart explanted from a transplant patient, and we kept it alive in this organ preservation system. We equipped it, and we can make measurements, hemodynamic measurements, as well as structural measurements on the tricuspid valve and actually the right ventricle here. We use mechanical test data. So here's a schematic of a tricuspid valve. We explant pieces of tissue and we do planar biaxial testing to understand its constitutive behavior. We use microscopy to understand the microstructure of the leaflet. So this is actually a 2-photon image in which we use a optical property called second harmonic generation to look at collagen in green. You can see elastin in red in the background. And you can also see the cells that we counterstained for their nuclei. And then finally, you've seen this model before. This is a finite element model, a high fidelity finite element model of a human tricuspid valve. And in red, we're highlighting the coaptation area, the contact area where the leaflets contact to make the valve function. And so we can look in these models for detailed reasons why in some patients the repair works or it doesn't. Our third thrust is on thrombus mechanics. So here, very importantly, we look at something called thromboembolic disease. You may have heard of deep vein thrombosis, where you have blood clots in your legs and they may break off and travel in your lungs. When that happens, we talk of thromboembolic disease. First, you have a thrombus, it breaks off or embolizes and then gets stuck somewhere. It turns out that hundreds of thousands of Americans suffer from it and die from it every year. Again, this is a disease of the right side of the heart, the venous vasculature. And we don't understand it very much. And what's fascinating about it is that it's fundamentally a fracture problem. And I say this because you see here, the thrombus sits there on the wall and it's about to break off. That moment where it breaks off, that's fracture. And I'm a mechanical engineer. I'm trained in fracture mechanics. And so we use this kind of training, we use these kind of tools to understand this better because very little, again, is known. And so we combine in vitro, in vivo, and numerical testing, as well as imaging, to change that, to fill these gaps in our fundamental understanding. What do we use? We use mechanical testing. Here you see what's called a mode 1 fracture experiment. So we took actually cow blood here and we coagulated it into this geometry. And then we use a uniaxial tensile test where we separate these two faces and look at the fracture phenomenon, the crack propagation, to understand what's going on in there. We actually do this under imaging, and we use various imaging techniques. There's actually a scanning electron microscopy image of a thrombus. You see here this fiber structure, it's called fibrin. You see red blood cells. So we relate the mechanical properties to the structure. To kind of capture this fibrous nature, we do something called discrete network modeling. You see here, this is a computer model of the discrete network. We can couple that to sort of a standard finite element model to understand how do these interact. And we do standard finite element models. So you see a finite element model of this mode 1 fracture experiment that you've just seen. Real quick, we also have other projects related to medical device evaluation. So we look at a device called an annuloplasty device. This is an image from a paper that we just published where we looked at a number of different devices and actually structurally understood them and mechanically tested them to compare them to each other. We do also prototyping where we look at how can we improve these devices because, again, they're actually not working very well. And we do in vivo as well as in silico testing. So we build computer models in which we can test these devices. All right, so the big question now, of course, there's so many great labs to choose from. This is an amazing department, an amazing university. So why should you choose my lab? First of all, you'll choose a supportive, understanding, successful advisor. For me, it's incredibly important that I support you as much as I can. And so when you join my lab, we talk about your future goals, we talk about your ambitions, and we make sure that I get what I need, which is you to solve important problems with me related to my lab's vision, but also that you get what you need, which is to learn your tools and to learn the programming language or equipment or experimental techniques that you want to know for your successful career. You'll join a fun, productive, and collegial lab group. So in my lab, we have really developed a very supportive peer-to-peer education and support system in which you really are never alone. You always have somebody who's on your side who will train you in the techniques and knowledge that you need to be successful in this lab. You'll learn tons of techniques, software, protocols, and equipment. We have a very diverse portfolio of skills, and you can learn whatever you want in the lab, and we'll be there to teach you. And I think most importantly, you'll make a real difference by tackling important health challenges. I choose the subject to the vision of my research because there are real problems that are not being addressed sufficiently. And so that's what we do. And I believe that I can make the biggest difference that way, and I believe you can as well. And so finally, of course, the big question, what are the next steps? I think, first of all, check out my lab website. I try to keep it as much up to date as I can. So you learn about papers we've published, our vision, my CV, my lab mates, information about them, etc. Follow me on Twitter because frankly, I just need more followers. But you also learn about me and sort of, what I think about and maybe also what I find funny. Check out my Google Scholar profile. So here you see our latest work. And you see that we've been incredibly productive. We're a small group right now, but we're young and we're a really ambitious group. So this is a great place to be if you want to be productive, if you really want to make a change. And then finally, I would love to hear from you. So please take my email, send me an email, and we'll make a Zoom appointment and we'll talk about, what are your worries, maybe what are your hopes, what are your visions, and I can share with you what mine are, and then we'll find that you will be a great fit for my lab. Okay, with all that being said, I really appreciate your time. Thank you very much for listening. Thank you very much for watching this video, and please shoot me an email. Really appreciate it.